to the webinar at Believers Church Medical College and Hospital. 2.5 crores infected, 8.5 lakhs dead. In India alone, 3.5 million infected, 75,000 getting infected every day, 62,000 deaths behind us so far, 1,000 deaths per day and counting. Yes, we are in the middle of a very dangerous pandemic, and it's very pertinent that we learn about the genomic pattern of this dangerous virus, SARS-CoV-2, if we have to be successful with contact tracing, diagnostics, treatment, development of vaccines, etc. So we have with us Scientist C at G.N. Ramachandran Knowledge Center for Genome Informatics, CSIR Institute of Genomics and Integrative Biology, an adjunct professor at the Indraprastha Institute of Information Technology, Delhi. His core scientific areas are computational biology and genome informatics. His major research areas include computational biology of non-coding functional RNAs, clinical genomics, precision medicine, genome informatics, organization of information in genomes, integrative analysis, community annotation of genomes, and large genomics data sets. He is an editorial board member of the Journal of Translational Medicine and many other peer-reviewed and indexed journals. He was awarded the Fellow of the Royal Society of Biology in 2017, BioClues Innovation Research and Development Bird Award in 2014, CSIR Young Scientist Award in 2012, and has won many other accolades. According to an excerpt from The Wire, Vinod Scaria's lab was one among the Indian labs which pioneered in initiating projects to analyze the SARS-CoV-2 virus's genetic data. He finished his MBBS in Calicut Medical College and went on to do his PhD from University of Pune and has trailblazed ever since. Dear Dr. Vinod Scaria, it's a great pleasure and honor to have you with us for this very interesting webinar on genome pattern of SARS-CoV-2 in India. Over to you, Dr. Vinod Scaria.
Thanks, Dr. Jacob, for the invite and for organizing this webinar. It's actually a pleasure to learn about the institution and the wonderful work that we do. So let me start my presentation. I think it's visible. Is the slides visible?
Yes, it is visible, Dr. Vinod.
Yeah. So over the next 30-35 minutes or so, let me discuss what we have been working in this area and what we did find from looking at the SARS-CoV-2 genomes from India. Before I start my talk, let me first introduce my lab. Sorry. Yeah, so I'm trained as a medical professional and later as a computational biologist. And what we do in the lab is to look at genomes on computers, and we call this a field which is known as genome informatics. The variety of things that we do on genomes, a part of the lab works on the basic science aspects of looking at genomes, which includes looking at non-coding RNAs and looking at the functions of non-coding RNAs. We try to understand how vertebrate genomes are organized and how they are functional. And another significant part of my lab works in the area which is at the interface of clinical medicine and genomics, where we try to work with a number of clinical counterparts from across the country as part of the Guardian network to look at patients who have undiagnosed rare genetic conditions. We use genomic approaches to be able to identify the mutations and in many cases be able to allow them to have a better life and a better family. A very small proportion of my lab used to work in pathogen genomics, and we started it almost 10 years ago. Was mostly out of my interest in microbiology. Was never been a big group anyway. We used to work on a variety of viruses, starting with HIV, trying to look at microRNA targets for HIV. Then we ventured out into looking at mycobacterium tuberculosis. And of course, when the pandemic struck, we were just probably at the right point, right time to be able to look at genomics of SARS-CoV-2 at a very large scale. And of course, you could learn more about the institute where I come from, that's the CSIR Institute of Genomics and Integrative Biology, and the research activities from the link out there. Now, I'll not spend a lot of time trying to introduce the virus because I'm sure you're all well aware about what SARS-CoV-2 is. Just to be brief, it has a RNA genome, a single stranded RNA genome of around 30,000 base pairs. It's a quite large genome compared to many other genomes out there, viral genomes out there. Now, what we all know about the SARS-CoV-2 genome, thanks to the researchers from China who put up the first genome of SARS-CoV-2, which sort of created the framework for looking at SARS-CoV-2 genomes on a large scale. Now, there was something very significantly different in this approach that the Chinese collaborators, the Chinese counterparts have done. Something very distinctly different from what of all microbiology that we have learned. In typical microbiology, that is clinical microbiology that we all learned as clinical professionals or medical professionals, the basic concept is to actually approach the patient, obtain a sample, and then probably go back to the lab and culture them using appropriate culture media. And here the problem lies because there is no one single culture media for all organisms. And you would readily appreciate this because no microbiologist would put up a culture without knowing what's the clinical suspicion. Now, that is significantly what genomics have changed, because if you really go back to this paper and follow the methodology, what it did was something starkly remarkable. They took the bronchoalveolar lavage from a patient, they did a total RNA extraction, did a ribosomal depletion, and straight away went to sequence. So you could understand that you could circumvent the entire process of culture and propagation of the virus. And this is remarkable because one, it reduces the time from the sample to the diagnosis. Second, it reduces the uncertainty in the approach for culture. And third, it becomes a low risk because, of course, culture propagates the virus, which requires very significant containment. Now, this is not something that was new. This approach of direct sample to sequence was available for quite some time, at least 6 to 7 years now, but it has never been applied so rapidly to look at epidemics and pathogens across epidemic. And of course, after the sequence gets generated, it goes through a detailed process of base calling, sequence trimming, assembly analysis, and so on and so forth. I'll not delve deep into it here, but of course, through the next sets of slides. Now, what has also happened in genomics in the meantime, in the last decade or so, has been a remarkable expansion in the scale and throughput to be able to do genome sequencing. And you would imagine the Human Genome Program, which is 3 billion bases, took approximately 10 years to completion. But today we can do genomes, not just one, probably dozens or couples of dozens of them, in one day, right, at a massive throughput that's practically available today. And this is exactly what made such an approach very possible, because you could take genetic material, whether it's DNA or RNA. Most organisms would have either one of these genetic material. And then, of course, you could fragment them into small fragments, and you could sequence these false fragments. And then, of course, ask the computer to be able to assemble these back into the sequence, which is sort of reflective of the organismal genome. And this convergence of technologies, one of high throughput sequencing, second of the computational approaches to be able to assemble genomes at high throughput, and third, approaches to be able to approach sample to sequence, has all converged in a very fantastic way to make this possible. Now you might ask, why would people do genomics of a pathogen? Because you know it is SARS-CoV-2, why sequence it again and again? And that boils down to the philosophical question of how organisms evolve. Practically all organisms that we know today evolved by accumulation of genetic variants, and genetic variants supposedly do occur at random, but not necessarily at random. And as they occur over ever generations, at some point of time, some organisms get a mutation or a genetic variant which confers them a selection advantage, and then they propagate further. And that's what happens to practically all viruses, all organisms, including ourselves. Now, what is really different between organisms is that the rate at which they mutate, or the rate at which they can accumulate genetic mutations. And to put a number to this, SARS-CoV-2 approximately, being an RNA virus, approximately mutates at around 37 mutations every year. That's approximately one mutation every 10 days or so. Now, looking at these mutations, and since you know what is the rate of these mutations, you can also track how the epidemic has progressed over time and over across geographical boundaries. And that is essentially central to what I'm going to talk about in the next 20 minutes or so. So you just need to keep this in mind, that the virus mutates at approximately 1 in every 10 days or so. And you would also know that a virus transmits from one individual to another individual in anything from 5 to around 7 to 10 days. So that means that every 2 individual or every 2 transmissions, you would accumulate 1 mutation. So that is the forensic fingerprint of how the virus mutated and evolved. To just give you an example, I will put this in perspective. And the first genome of SARS-CoV-2 was collected from December 26th, right from Wuhan, one of the early patients who had SARS-CoV-2 infection. And by January 8th, it had an additional mutation from the original sequence that it had. And in February 27, another patient from Guangzhou had 3 other mutations from this original sequence of SARS-CoV-2. And this is how the virus evolves over time. And this essentially gives us an insight into how is the virus mutating and how is the virus moving across different countries in the world. Now, the other remarkable thing that has happened in this epidemic is that it is not just one virus sequence that is available, but today, as we stand here, we have close to around 80,000 genomes of SARS-CoV-2, thanks to researchers who have collected, who have processed these samples and sequenced in their labs all throughout the epidemics from different parts of the world. And most of this data, thankfully, is available in open repositories like the NIH Open Repository, which is Scaria of the GenBank, and a database which is called GISAID, which used to previously host information about influenza virus sequence. And of course, there are some countries which have gone into massive scale genome sequencing expeditions, like for example, the UK has the COVID Genomics Consortium, which has practically sequenced around 30 to 40,000 genomes of SARS-CoV-2 spread across different regions and different time points in UK. Now, while these datasets are out there, it's very important to also create resources to be able to search and retrieve these information, which is scattered across multiple resources. And that's when, way back in early April, we put together this resource, which is called GenomePDM, which allows users to search through this information and be able to retrieve the isolates that you would want to retrieve for very specific analysis. And all these initiatives have been fueled by a lot of volunteer participation of students, a lot of citizens who were interested in contributing to a novel endeavor and then therefore contributed to building these resources. Now, the other thing that you need to keep in mind is that once a virus evolves, and all viruses, I would say, would evolve, they would make it a bit different from each other, and these differences can be represented in what we call as a phylogenetic tree. And a phylogenetic tree essentially is a trace of the genetic lineage of the virus. Now, when you say SARS-CoV-2, that necessarily means it is a new strain of coronavirus, distinctly different from SARS-CoV, which was essentially the first SARS epidemic in the world, pandemic in the world. Now, between these strains, of course, there are major differences, so they would be very different in their branch points. And within the strains, so for example, SARS-CoV-2, I said there are 50,000 odd isolates. Each of them would be different from each other by a few genetic mutations. So they would be closer together in the branch points. And this is what you need to keep in mind, that you can trace a species, you can trace a strain, or you can trace clades or genetic lineages depending upon how close or how distinctly different these isolates are from each other. Now, if you put the entire data of SARS-CoV-2 genomes, which is available across, and this has been done by a lot of labs across the world, and this is one representation by this database called NextStrain, which cataloged data sets from all across the world. In general, the isolates from all across the world could be divided into 10 genetic lineages or clades. And this is one of the sort of representation of these 10 genetic lineages. And they can largely be divided into two super clusters or super clades. One you call as an A superclade, which is mostly European, and the B superclade, which is mostly Chinese and Southeast Asian. Now, the first genome of SARS-CoV-2 essentially belongs to the B superclade, which further got divided into B1, B2, B4, so on and so forth. And of course, the A superclade was the out of China isolates, which are mostly from Europe and adjoining areas as spread across the world. And A2A is a predominant isolate, mostly from Europe. This is just to keep in mind what the different indications out there. I'll come back to the slide again. I said before that the virus mutates, and the virus mutates at a very constant rate. And then therefore you should be able to use this molecular fingerprints of genetic mutations to be able to understand how did the virus evolve and spread across different parts of the world and across the different time points. And that's what we call as molecular contact or ancestry tracing. So in other words, if you get a patient with SARS-CoV-2 early in the epidemic, suppose we were asking this question in March, right? And if a patient in for example, Kerala got an epidemic, got an infection with SARS-CoV-2, you could sequence the virus and ask this question: where did this guy come from, right? So if the person was from Wuhan, then you would get a virus which belonged to the B clade of the phylogeny. If an individual was from Europe, you would get from the A clade or the A superclade. Mostly in this case, it would be A2A. And that's exactly what had happened. The initial people who got the epidemic infection from Wuhan and traveled to Kerala did belong to the B cluster of the phylogenetic tree. Now, through the epidemic in India, mostly through March and April, people also have moved across cities, right? From one major city to another major city, or from a major city to smaller cities just after the lockdown. And I'm sure you would have seen this in news. So in many ways, you will also be able to track that spread of the virus, because you can look at the fingerprints of these viruses isolated from across the country and be able to ask this question: where did this guy get this infection from? So in other words, you should be able to look at each state, each region, each district, and whatever smaller number of levels you can go to, right till family, to be able to understand what is the relative contribution of people traveling into the country and traveling between different cities in the country to be able to get to this level of the pandemic. And this is very remarkable also because different cities in the country would also have different proportions of the clades of the phylogenetic classifications, depending upon where they got the infection on the first place. And this is an old map, mostly drawn early August. This sort of shows how the infection has come into the major cities, and the major cities here are, of course, Mumbai, Delhi, Kolkata, Hyderabad, and of course Bangalore. And then how has this infection spread across major cities and from major cities to minor cities and so on and so forth. So fortunately today we are in a fortunately good position that we have close to 2,700 virus genomes as of today. I'm sorry, this is an old slide. And this comes from around 21 major research and clinical organizations and covering almost all major states, that is 21 major states and union territories. And the largest number today has been deposited by one organization, which is the Gujarat Biotechnology Research Center, which has deposited close to around 500 genomes of SARS-CoV-2 in public domain. So that is a unique resource that we can have access to be able to understand what is happening in the country. The obvious question is, why do we need to understand genomes? One, you can understand the evolution of the virus. You can understand the origin and the molecular trace of where did this virus come from and how did it reach there. And third, of course, you can look at community outbreaks. And this is a very pertinent question because you could get outbreaks, and you see this in news almost every time, that you could get an outbreak from a travel together, for example, in a flight. You could get an outbreak, for example, from a hospital. You could get an outbreak from a meeting, so on and so forth. And now you'll be able to understand and trace and say, where did this virus come from? Did it come from two different individuals? Did it come from one individual? Did it spread as you suggest, or did it occur independently? And of course, more importantly, looking at mutations will also allow us to evaluate diagnostics and also probably evaluate vaccines and therapies as they come. We don't have them yet, but when they come. So if you now go back and look at all these isolates from India, for the last 3 odd months, India has largely had 6 plus 1 clades. I would mention 6 because these are 6 of the 10 clades that I sort of mentioned before. And we have one clade which is very unique or distinct. I'll not say exclusive, but unique and distinct, that is found in India. And we named this clade as I, or the A3I clade. I because it's a dominant clade in India and not dominantly found elsewhere in the world. And A3I because it actually bifurcated from the A3 clade at some point of time. Now, incidentally, the A3 clade is also found in India, and A3 clade in India traces its origin from travelers from Iran who came into mostly the northern states, Jammu and Kashmir. But nevertheless, the A3 clade is not, as you would imagine, diverged in India out of these travelers, but it had diverged out much before it had entered India. So in other words, it is very similar to A3, but has diverged much before it entered India. Now, you could also browse this resource if you have time and you have interest in this particular field. There is this resource that we have built where you can browse almost all those genomes, today around 2,100 odd genomes, and be able to click or mouse over each of these genomes and understand very pertinent information about these viruses. Now, when I say there are clades, you also need to make it important that these clades are essentially genetic lineages, so they would also have a genetic fingerprint, which sort of defines this particular clade. Now, for each clade, this could be one or more mutations, which can pretty much define the identity of that particular clade. In other words, this is important because going forward, when you need to do large-scale genetic epidemiology, you would not probably want to do sequencing of a large number of isolates, but now be able to look at just a few variations which sort of encompass or which can classify these viruses into different clades. Now, to give an example, A2A clade, which is a blue one out here, is a majority clade across the world and also across India. And the I, or the A3I clade, has essentially 4 genetic mutations which also define this particular clade, and some of these mutations are also shared by the A3 cluster, as you would see here. So this is how you can actually classify each of these viruses into different or distinct clades. Now, I said the virus mutates over time, so we were very interested in asking this question, whether the viruses mutate all in the same rate or different rates of mutations. And if you take an average of all these isolates out there, the virus mutates at approximately 3.7 per month. That's approximately once in every 10 days or so, approximately, right? But now if you ask the question, does different clades of the virus also mutate at different rates? The answer is probably yes. the A2A, which is a major cluster, of course, mutates at an average, close to average rate, and that's understandable because that's a large number. But you would also notice that the A3I clade actually mutates at a much lower rate. It's 1.19 into 10 to the minus 3 per site per month, compared to 1.48. So this is something that is different and significant statistically, but we don't really know why this happens. Now, you could also go back and ask this question, different clades that I sort of mentioned, that largely two dominant clades which account for majority, I would say 80%, 80 to 90% of all the isolation there, which is the A2A and the I or the A3I. The A2A, as I stated before, are from travelers who have traveled from Europe, and I is sort of indigenous cluster of viruses, which doesn't find a dominant sort of pattern elsewhere in the world. Now, different parts of India has different proportions of I and the A2A. And for example, if you look at Gujarat, there's a majority of A2A. But for example, if you look at Delhi or Telangana, it has a majority of the I clade. Now, what we really do not know is what are the clinical implications to this, which we are following at this moment. But what we know is the I clade essentially came into India from a single or a couple of individuals and spread across the country through a super spreader event at a distinct point in time. And we can also calculate what approximately is this point in time, and it is estimated to be sometime in end of February to early of March. And from that single super spreader event, it has spread all across the country, and different states have different proportions of these people who come from the super spreader event, and then therefore have different proportions of this virus. And we could also trace the dynamics of these viruses over time. And what we show here is that the peak of the I or the A3I epidemic was sometime in March and April, and then it has weighed down and get slowly replaced in proportions with the A2A clade, which has now become the dominant clade out there. And today, of the isolates that are available in the country, close to around 70% of the isolates belong to the A2A clade in total. And approximately around 18% of the isolates belong to the I or the A3I clade. And this was from a 50% I A3I at the beginning of April. So you could see how the dynamics has changed for these clades over the time. Now, what we really do not know is how these clades are different in their pathogenicity. But of course, having said that, there's some evidence which has been emerging in global literature. And one of this is a particular genetic mutation, which is called the D614G. And this is one of the mutations which sort of mark the A2A clade. Now, a number of researchers have suggested with experimental evidence to suggest that D614G, which is essentially a mutation in the spike protein of SARS-CoV-2, allows it to bind more effectively to the AC2 and TMPRSS2 receptors in humans, and then therefore results in increased infectivity of this virus. And of course, there have been now reports of other mutations, for example, in the ORF8 gene. Either this could be deletions or single nucleotide mutations, which can cause attenuation of the virus, or in other words, create a much less severe or an attenuated state of this virus. What we really do not know is how this difference in the immune response is contributed by this virus. But now, of course, you would have also heard about reinfections emerging, at least in case reports. And these are essentially because of mutations which are significantly different from each other in probably eliciting the immune response. And of course, there are a lot of biology that needs to be understood, and a lot of more work that needs to be done in looking at, for example, how does a particular clade have a much lesser mutational rate compared to others, and so on and so forth. Now, for all of you who are not really into genomics and into analysis of genomes, all this data is now compiled into a searchable, interpretable format that is now available in this resource called Indicos. This today indexes close to 2,700 odd isolates and close to around 3,000 genetic variations. And all these genetic variations have been annotated for a variety of features, which includes where these variants are prevalent, whether they are in a particular protein domain, does it change the immunogenicity of the protein, does it fall into, for example, the diagnostic primer sites, are they conserved or not conserved, so on and so forth. So this information is constantly updated, and made available to the research community out there. And probably this is the only resource of its kind in the world which sorts of annotates genetic mutations for SARS-CoV-2 and provides the functional annotations. And of course, having said this, there's much more things that need to be done. And the first and most urgent thing is, can we use this information to build better diagnostics? Can we also use this information to design better vaccines in terms of understanding the immune epitopes and how genetic variations could change them? And of course, in basic biology, trying to understand what are the functional consequences of these mutations, and how can we actually understand the biological system through these basic processes out there. And what's really more important is, how can we build better diagnostics? Now, the state of the art today, or the gold standard today for diagnosis of SARS-CoV-2, is using a technique called real-time PCR. Now, PCR, or polymerase chain reaction, depends upon the concept that you have oligonucleotide probes, or DNA probes, which can identify and bind very specifically to regions in the viral genome. And this amplified product is probed using a fluorescent labeled oligonucleotide, and that is what the signal that you sort of measure. Now, having said this, you should also realize that these are all based on hybridization of DNA or RNA each other, and this is sequence dependent, and then therefore genetic mutations in the sequences could affect how the probe binds or how a primer binds, and affect the efficacy of a particular genetic assay. And then therefore there's an opportunity to be able to map these genetic mutations in viruses and ask a very simple question: does these mutations occur in the primer binding sites? And if they do occur, can we make better diagnostics which can sort of get rid of this phenomenon? And this is a study that we did almost a couple of months ago, where we compiled close to around 50 primer and probe sequences, which are all available in public domain, and asked this question for the Indian strains or Indian isolates out there: can we see what is the frequency of mutations that occur in these sites? So the red ones out there are the variations that occur in the primer binding sites, and the y-axis is essentially the frequency of this event occurring among the 1,000 odd genomes out there. Now, what you see is there are, of course, a few genetic mutations, mostly in the N gene, which are high frequent, and some of them which in the ORF1ab gene, which are also at modest frequency. So we now know that those sets of primers and probes are probably going to give a false negative report because of the fact that they would perform in a suboptimal fashion. And this is also an opportunity, because now you can sort of design probes into regions which have low frequency of variations, and then therefore low chance of figuring out in a position which has a lot of mutations. So this is one thing that we're currently validating in terms of building better reagents for diagnosis of SARS-CoV-2. And of course, the obvious next question is, while we say that we can do 2,000 or 2,700 genomes in the last 4 months, it's really important is how can we extend this into a state-of-the-art or a gold standard for genetic diagnosis. And that is when one of my colleagues, Dr. Sridhar, had this concept of creating what we call as a mega lab. Now, a mega lab is essentially the idea, the premise of this mega lab is, how can you use sequencing for detection of SARS-CoV-2 as well as for genetic epidemiology, because you can also do detection of SARS-CoV-2, at the same time also be able to do the genetic epidemiology in terms of tracing the mutations and the spread of the virus. And what we have used here is essentially a test or the assay system which is called the COVID-SIC, which is an FDA-approved reagent. And we are fortunate to be one of the first real-life application of these assay system anywhere in the world on clinical samples. And of course, this was made possible with a wonderful group of friends and colleagues at IGIB, and of course, a fantastic collaborator, Dr. Sridhar. Now, the idea of this is very simple. All these samples were collected through a self-collection. So you have tubes where individuals could self-swab themselves and, of course, put an ID into an app. And these samples were processed for RNA extraction, were sequenced, analyzed, and could be reported back onto the app. And this was part of a large-scale surveillance program, which today has crossed around 1,500 people, and you can do around 3,072 results in 12 hours. That's a scale of activity that can happen. So you could imagine this is a number that is almost impossible to be attained by a single PCR or RT-PCR machine. But today this is possible by using high throughput sequencing at a cost and time scale which is comparable to RT-PCR. And what we also show in this entire approach, I'll not delve deep into the slide, through this approach is that we have a much better sensitivity for detection of SARS-CoV-2, at least with a gain of around 8 to 9% of clinical detection of SARS-CoV-2 compared to RT-PCR. And if you have been doing RT-PCR for SARS-CoV-2, you will also get into classification which is inconclusive or sometimes sub-echo, because the two probes that you use, either of them could be positive and the other could be negative, and then therefore become an inconclusive result. Since you sequence the genome in entirety, there is nothing called inconclusive. You can either call it as a positive or you can call it as a negative. So you can get rid of the inconclusives, and in our experience, 75% of the inconclusive could be assigned as positive. So that is a significant gain in terms of, one, the throughput of testing. You can do 3,000 sequences in a single day. Second, in terms of the sensitivity for detection with a clinical gain of around 3 to 9%. And third, to be very accurate in what you say, in trying to avoid inconclusives or conservative or positive. And of course, there are many more programs on SARS-CoV-2 that we have been running and trying to look at better diagnostics, trying to look at better ways of looking at epidemiology, and trying to trace the epidemic. And of course, a number of approaches to be able to connect to patients and be able to understand very crucial aspects of the clinical progression of the disease. I think I'll stop there and take questions, but before that, let me acknowledge a lot of people who made this possible. This includes a lot of people in the lab who have been working from home and contributing to a number of resources, datasets that has been out there. A wonderful team which put together the COVID-safe approach in the lab, mostly with Dr. Sridhar Chivasubu, who has been a long-standing collaborator in many different programs. A wonderful set of clinicians from across the country who made this possible, and also systematically worked towards collecting clinical samples from across the country. And of course, this is all possible with a lot of clinical as well as non-clinical research collaborators from across the country who contributed genome sequences of SARS-CoV-2 in public domain. And of course, all the funding for this entire activity came from CSIR through fast-track grants, which made this all possible. I think I'll stop there and take questions. And while I do that, of course, there's an app that we have created which sorts of enables connecting patients, families, clinicians, as well as researchers. And if you're interested, probably you could try out this app. Thank you. I'll take questions.
Thank you so much, Dr. Vinod. That was indeed a wonderful session on various genomes of COVID-19 in India, especially, and we were really fascinated to know that there are more than 2,700 virus genomes, and especially the A2A being dominant in India. These are all new things which we are learning, and the concept of mega lab really was fascinating. And thank you so much for this wonderful exposition of what is happening in the genomic world in terms of COVID-19. Thank you so much. So, yeah.
So if there are any questions, I would be happy to answer them.
Yeah. You were talking about this mega lab, right?
Right.
So, can you elaborate a bit about it?
Yeah, so a typical RT-PCR with a sort of a full-time workforce could do anything from around 96 to 384 at the max of samples per day, right? And especially for states like Kerala, which is going to see the peak of the epidemic happen, right? You need to scale up the testing capacity, and this testing capacity is not easy to scale up because it's not just machines. You also need people. You also need the collection protocols. You also need the reporting protocols to be in place. So there's a lot of logistics involved in the scaling up. Now, the other problem in scaling up is that you need the extended capacity only for a very small period in time, right? That is pretty much during the peak of the epidemic. And typically looking at the peaks in India, this is anything from 1.5 to 2 months, right? So you don't really require to buy in a lot of equipments to just handle that 1.5 to 2 months. The idea of this is that can we repurpose sequencing equipments which are already there, right? These are high throughput sequencing equipments, and can we use these equipments to be able to support the scale up requirements for testing? And that's the entire concept of this assay, that you can scale from anything from 300 samples to 3,000 samples in 24 hours using these equipments. So while you don't require that scale, you can still do 300 samples. While you require that scale, you can do 3,000 samples in a day. And this is the kind of capacity with just one equipment that you can build, right? So the idea was, how can you build that kind of an assay? Now, the consideration is, one, it has to be in a time scale that is comparable to RT-PCR. Second, it has to be accurate, if equally accurate to RT-PCR or more accurate to RT-PCR. And third, of course, by doing this, provide additional information on the genetic epidemiology, which is not possible with RT-PCR today. RT-PCR today is only a positive or negative report. It doesn't tell you which clade or which family or lineage did this virus come from. So the idea of building this mega lab was to be able to do all three of them, right? To be able to do testing, to be able to do testing accurately at scale, and third, to be also able to do genetic epidemiology at a price that is comparable to RT-PCR. So this particular assay today can do the sequencing at practically the same cost that you would do an RT-PCR.
Thank you so much, Dr. Vinod. In keeping with the fact that the virus is mutating continuously, how frequently do we have to change the DNA probes used for testing?
So in many ways, you have already been changing DNA probes for testing, because the early reagents that came into India during the early epidemic mostly tested for the N gene, single gene. And meanwhile, somewhere in March, April, ICMR testing guidelines changed it to 2 genes, which is the N gene or the E gene plus DRP. And what we today know is that many of the early reagents that came into India are probably not working as efficiently because of the genetic mutations. So now there are new probes and new reagent sets that have come acting into this, right? Now, in many ways, this change didn't happen by looking at the genetic variations, but probably happened because of the inefficiency in clinical settings that was reported, right? And the real big problem in clinical reporting is a false positive is still fine, because at the max you will quarantine that person. But a false negative report will mean that an infectious individual is going to roam around and spread the infection. So you need to have more specificity to this testing. Now, the idea is, now once the positivity goes down, right, then you will need more accurate testing. So I'll put it in this perspective. In the peak of the epidemic, anybody who has fever, cough, right, you would anyway brand him as COVID and test and treat him as COVID, right? And you might probably not even wait for the report to come. All the precautions would already be in place, assuming a priori that the individual is positive, because everybody else who has that symptom is COVID positive, right? Now, early in the epidemic and late in the epidemic, you would need a test which is extremely sensitive, right? Because you cannot afford to miss one individual who is positive back into the community, because that's going to infect more people. But that's not the consideration when you have at the peak of the epidemic, where the consideration is that if you have the symptoms, probably you have the disease, and therefore they will treat you, right? So once the epidemic goes away, once, for example, the air travel and industry travel picks up, you will need a much more sensitive assay to be able to do this. And you will also need to do it at scales which are not been there before. And that's essentially the consideration. And then therefore, you would need better assays, better probes to be able to do this.
Dr. Vinod, we have ramped up testing in our country in the last few months, but would you say that very early levels of high testing would have brought our infectivity to a much lower level?
There is something called an ideal situation and something that is realistically possible, right? So, yeah, speaking idealism, theoretically, if you had tested all air travelers into the country early on, right, you could have prevented it from occurring, in theory, right? That's in theory, right? It may not be practical. And at least in theory, if you had tested everybody who is traveling, you could have prevented the infection. Right? But that's not practical, right? So, now having said that, we always have to balance between theory and practicality, and also be able to use our resources to the best possible use. And I think that is the aim of every government, right? It is not just our government, but practically every government, is to consider this in terms of striking this balance between the resources that you have and the resources that you can best utilize.
Thank you, Dr. Vinod. One more question. Now, we spoke about the super spreader, which came to India first. So, can we trace it back even before the super spreader, as in where the super spreader came from? Is that possible?
Yes, so there are 4 genomes in public domain that has been there recently, and these genomes mostly came from Southeast Asia, and at least one from an African country. So we approximately think that it is one of the travelers who came from Southeast Asia. By association, we have of course no proof to that.
So now everywhere the buzzword is vaccine, right? So we have an interesting question. Is the Russian vaccine Sputnik successful or not?
You would never know until it goes into phase 3. And that's true for every vaccine, right? You would never know. Now, there are a few considerations that you also need to keep in mind. That the vaccine is to produce an immune response, right? And what we do not know is whether this immune response is going to be protective or not. Vaccines will anyway produce immune response. What we need to see is whether they are protective, because there's a significant difference between non-protective immune response and protective. And third, there's also a balance to be striking in this particular case, because at least in COVID, there is this concept called antibody-dependent enhancement, right? That just means that a lot of antibodies in excess are going to create more problems. So that's exactly why we need to always be treading cautiously in building a vaccine, because one, it needs to elicit immune response, it needs to elicit a protective immune response, and third, it should not induce harm to the individuals who are taking it. Right? And that's why much longer periods of studies are required to be ascertaining what is the optimum balance. And at this moment, we are still clueless, and I think time will tell us.
And talking of these clades, A2A is the current dominant clade, right? Why did this transition occur? Initially, it was A3I, and how come this transition to A2A? What was the reason behind that?
So there could be multiple reasons. We don't know what of these reasons are right? One of the reason is that since A3I was an initial sort of an outbreak event which spread across different states, right? Most states early in the infection were all prepared to be able to test individuals, quarantine individuals, and be able to offer the best of care, right? Like we always used to joke that the best of care is almost always in the beginning of the epidemic, because you are this one VIP patient who has COVID, and the entire hospital looks after you, right? And that doesn't happen at the peak of the epidemic, because then there are hundreds, if not thousands, of people who are vying for that one single doctor, right? So there's always a disparity in care that occurs during the period of the epidemic. So since I happened through a super spreader event, and it has spread to almost all states, and almost all states were at that point of time actively pursuing contact tracing and testing. Probably that is the reason why it could have come down. That's one possibility. The second possibility is that probably it was a virus which is not very infectious. It would have spread through a super spreading event, agreed, but then probably it was not as infectious to spread further and then therefore died down. That's possibility number 2. Possibility number 3 is that A2A, which has a D614G mutation, is anyway known to be more infectious. So it could be just a competition between normally infectious isolate and extra infectious isolate, or super infectious isolate, and then therefore by numbers a super infectious isolate would win. So there could be multiple different possibilities. Now, frankly speaking, we don't know why this happened. These are all possibilities.
That's interesting. Thank you, Dr. Vinod. One last question. Drugs, remdesivir and tocilizumab and so many other drugs are now doing the rounds. So does this genomic mutation has anything to do with response to drugs?
Very interesting question. So yeah, like with vaccines, the problem here is for none of these drugs we do have concrete evidence to say that it works, right? And even if it works, we don't have concrete evidence to say that how does it work. Now, once you have a drug and we know how it works, then of course we could go back and ask this question: does genetic mutations change how it works? Right? Until that point of time, we are still in the dark.
All right. So I think we'll close the session if there are no more questions.
It's a pleasure. And if you have more questions, of course, you could email me or just write to me.
Definitely. So thank you so much, Dr. Vinod Scaria. It was a great honor to have you on board for our webinar. So a big thanks from the Believers Church Medical College Hospital community. Thank you so much. Looking forward to collaborate with you in a lot of research and also in future webinars. Thank you so much.
Thank you very much. It's always my pleasure.
Thank you, Dr. Vinod.